Hey guys, we're going to pick up where we left off in lecture, and we left right up here with immune tolerance. I went ahead and covered this anyway, but I said I'd come back to it. So, to reiterate just real quick where we were talking about last, we were talking about how in the proliferative stage, we end up with 500 genes that can be rearranged to create unique lymphocyte receptors. We start out with random receptors, and then ultimately some of those are selected by antigen, and that's where the clonal selection comes in. So we see lymphocytes developing into unique combinations. We end up, we'll talk about this part here in just a second. We end up with mature lymphocytes here, each with a unique receptor. Then in the next stage, at some point, an antigen will actually trigger one of these random lymphocytes. That particular lymphocyte then will be said to be clonally selected, meaning the antigen binds, and then expansion, meaning that it will now make copies of itself. And this is where it'll divide to make copies of both the memory cells and the effector cells that I mentioned. Okay, that detail is not really given here, but that's what will be happening. So that is the idea behind the clonal selection and expansion. But remember that it starts with random gene combinations creating random receptors. This figure here is attempting to show the gene connection, or the gene receptor connection, I should say. So here it's showing different colors of the DNA representing different genes, just showing a small subset of the 500 different genes. And then it's showing how these different colors, again representing the genes, can be rearranged in different combinations. And then those combinations, those combinations lead to different receptor configurations. So every time you switch these genes around, the equivalent of switching the colors around, you get a different receptor with a different specificity. So that may not make perfect sense, but that is the idea behind that. Now, immune tolerance is an issue that arises, or I should say one of the issues that arises is that when you randomly create receptors, one of the possibilities that will eventually happen is you're eventually going to produce receptors that target the body's own cells. So this process has a serious flaw in it, in that you're not creating receptors to match the antigens once they come in. You're just creating random receptors to begin with. And what will eventually happen is you'll create a receptor against your own body. So clonal deletion is a process by which we prevent that. And ultimately what's happening is before the cells become fully mature, they are essentially tested out, so to speak. And this happens during the development in the lymph nodes, or not in the lymph nodes, during development in the bone marrow for B cells and in the thymus for T cells. If any cell in that area responds too heavily to the body's own molecules there, then that cell is clonally deleted, meaning it's removed before it enters full maturation or enters into circulation. So just going back here, with this figure, that's what we're showing here. That as these develop, any cell that targets the self early on is eliminated. Now, there's a lot of theories on exactly how autoimmune disease works, but this is one of the more obvious ways that it's thought to happen, and that this deletion process doesn't work properly, or somehow that fails, and one of these cells gets out into full maturation that's not really, shouldn't be, I should say, and it ends up at some point targeting the body's own cells. So autoimmune is a very, very dynamic disease, and there's lots of different reasons why it's thought to happen. So this is not the only reason, but that's a note about one of the ways that the body avoids targeting the self, clonal deletion. Getting into the next part here, we talk a little bit more about the common way that antigens are actually brought to the lymphocytes. So recall that the lymphocytes are sequestered away in the lymph nodes and sequestered away in the spleen, primarily. These are body locations that are not open to the rest of the body systems. So in order for the antigen to effectively get to those lymphocytes, what actually happens is antigen-presenting cells, primarily macrophages, will ingest foreign material and then bring it to them. And this is where the second line of the immune system intersects with the third. So let me just put that here like we were on the whiteboard. Our first level is barriers. Our second level is the non-specific. Our third level is the specific. And what we're really saying here is that between the non-specific in the specific, we have antigen presenting cells that are going to bridge this gap. So APCs, antigen presenting cells, the most common being macrophages. So the way that antigen gets from the non-specific to the specific line is through antigen presentation. And what's key about that is that these macrophages are more or less ubiquitous throughout the body, whereas lymphocytes, like B and T cells, they are not. So these cells being anywhere can bring the antigen from anywhere to the specific regions where these cells are located. So that's the way the body kind of handles all different aspects, all different locations, and centers the necessary things, the antigens, back to the key locations. Now, this is not necessarily intuitive, but this is the way it actually works. So antigen presenting cells will ingest foreign antigen and travel to the lymph nodes and also the spleen, where these lymphocytes are located. Now, at that point, the macrophage, or whatever antigen presenting cell, will be displaying antigens on its surface. So they're going to ingest, phagocytize the microbe, break it down, and then literally display the antigens, various antigens, on the surface. And recall that a single microbe could have literally hundreds of different antigens. So we've got lots of different specific little unique antigen molecules here. These macrophages will all start migrating back towards the lymph nodes. Primarily, some of them will make it to the spleen, but most of the time they're going to the lymph nodes. And when they get there, they're going to start moving around the lymph nodes until they bump into a T helper cell that's got a matching receptor located on that cell surface. Let me pause it just real quick and see if I can find a good picture of the lymph node. Okay, so here's a figure I found. It's actually a pretty good one. It's showing a lymph node here with a cross section, and it's showing a antigen presenting cell, in this case a dendritic cell, very similar to a macrophage. Actually, that's a type of macrophage, if you recall. And it's moving in through one of the lymphatic channels into the lymph nodes, and it's bringing with it the antigen, and it's going to display that, and it's going to bump into a T lymphocyte that's going to activate this T lymphocyte, and then it's going to set off the expansion. So this is essentially watching here the clonal selection, where the antigen presenting cell brings the antigen in, it binds to a T lymphocyte with a matching receptor, and that sets off the expansion, meaning that cell now activates and divides and make copies, and it's going to go on to activate then the B cells, which will go on to activate further processes. So that's kind of a quick visual of what's happening there in the lymph node. Okay, so and this I put this figure back on here to show that this is what would actually be happening here in this entry of the antigen. So antigen presenting cells include macrophages. That figure there just showed dendritic cells. These are a type of macrophage, type of histiocyte. And then B lymphocytes can also do this, but that's a little bit more of an advanced immunology topic. So the idea is that any antigen presenting cell, mostly these two here, will display antigen, bring it in to the lymph nodes primarily, and then activate T cells, which then T cells will go on to activate further processes. So once we start activating these lymphocytes, we set off really the lymphocyte challenge, the third stage of development, or the third stage of the immune response. So this is going to primarily start off with helper T cells and cytotoxic T cells. Really, I should say, it's going to start off with helper T cells. Helper T cells are going to be the first cell that are activated by the macrophages. And the term helper T cell indicates that what they do is actually help other cells in initiating their reactions. So what they do is they end up activating B cells, they go on to activate cytotoxic T cells, and they even amplify the reaction by activating other macrophages, which will then now be stimulated to go back to the site of the infection and bring even more antigen back, activating even more B cell or more T cells, and then furthering the entire effect. So it's kind of a cyclical effect in which the response actually increases the response further. But helper T cells are key in that they're going to set off the reaction with all the other cells. So at this point, then they go on to activate some of the cytotoxic T cells. Cytotoxic T cells go on to destroy virally infected cells or compromised host cells. So compromised host cells would include virally infected, cancerous cells, or even damaged cells. So T cells do a lot of house cleaning, so to speak, and that gets into a different area of immunology. So in terms of microbial infection, they're primarily going to be targeted targeting virally infected cells. And they're going to be the primary mechanism by which those cells get eliminated. And if you recall from Chapter 6, that's important because once the cell is infected, it can't be saved. So you have to eliminate the infected cell so it doesn't continue to produce more virus. So here we're showing the lymphocyte response, which is different in T cells and B cells. So T cells have what's called a cell-mediated response, primarily involving the cytotoxic T cells, and that was what I just mentioned with them destroying the virally infected cells. And then B cells is what we're going to get into in more detail now, and that is going to be involving antibody production. So just to clarify here, my PowerPoints need a little bit of reorganizing. I'll be first to admit that. To clarify, the T cell response primarily involves cytotoxic T cells being activated and eliminating virally infected cells. I'm definitely leaving a lot of things out there, but that in a nutshell is what's happening. And I'm trying to keep that part of the topic relatively brief because there's a lot of complicating details there. So in a nutshell, that's really what they do. They're activated to destroy compromised infected host cells. And that's, in a nutshell, the cell-mediated response involving cytotoxic T cells. With B cells, we have antibodies doing most of the work. So when B cells come into the picture, they are typically activated by the helper T cells, and we call this a T-dependent activation. Sometimes, and just to give a little bit more detail on how this can happen, sometimes antigens will activate the B cells through a T-independent reaction. And this means that they can essentially activate the B cell directly. That can happen. It's not the most common way, but these are the two mechanisms by which this can happen. So sometimes B cells can just be activated independently. But either way, what ends up happening is the B cells start to proliferate and make copies of themselves just like the T cells did. So the expansion. Typically only when they're activated by the helper T cells. And one of the things that happens when the B cells start dividing and making copies is they're going to produce memory cells along with plasma cells. Now it's also showing regulatory cells, and that's another different aspect of the immune system. So coming in, once again, here's the microbe, here's the ingestion by the antigen presenting cell, activating the T cell. T cell goes on to activate a B cell. B cells now expand. We make memory cells. We make plasma cells, which are the effector cells of the B cell line. Okay? So ignore the regulatory cells. That's a more in-depth part of this. So now plasma cells are going to go on to produce these antibodies or immunoglobulins. And here you're seeing one of the more elaborate types that's got a bunch of different antibodies attached together. But the idea is this is essentially the B cell response in a nutshell. So antibodies, once they're synthesized by these plasma cells, they start to go on and have an effect against the microbe. And here they're showing one of the effects, which is binding to a virus and blocking that virus from attaching later. So the whole process of the B cell response revolves around these antibodies, and they look like this. This is a very basic antibody shape. They have a Y shape, as I've drawn and alluded to in class, and each side of this antibody has a binding site for the antigen. This is called the variable region, and it's called the variable region because it's different for every antibody. Each one is created by those 500 gene combinations and a rearrangement of them, and every antibody is a little bit different. So this is where the antigen is going to bind. It's going to have to match with that lock-and key specificity to that particular antibody. Each arm of the antibody is identical. So they don't bind two different antigens, they bind the same antigen, but they can bind two different antigens simultaneously. It's like having two arms that grab the same thing. So that's where the actual action against the antigen is going to take place here. Down here we have the constant region, and they call it the constant region because it is more or less the same in all different types of antibodies, so there's really no variation here. And this is this is going to be where the antibodies can make interaction with the body's own white blood cells. So for example, it says down here binding site for cells. This is going to be, for example, where they can interact with other white blood cells, which can trigger further reactions. Okay, so for the most part, our primary interest involves these antigen binding sites. Now this is obviously an illustration. This is a three-dimensional model here trying to show what they actually look like. So this is a three-dimensional space-filling model that accurately depicts rather what the antibodies actually look like. So they're very complex little things, and so that's why we typically show it in this simplified version, because it's much harder to actually see the connections with the binding sites and whatnot with this here. But that's a more accurate look of what they actually appear. So this is reiterating what I just said there, talking ahead of myself. So go ahead and copy that down. One more thing I should point out. Antibodies are basically a secreted receptor. So I've shown these little Y-shaped receptors on top of the cells. Drawing little bunny rabbits here, but I've shown them on top of the cells sticking out. This is basically the same thing, except rather than sticking out from the cell, it's secreted and released outside of the cell. So it's a secreted receptor, essentially. And in this sense, they become free-floating, and they travel throughout the body. And these B cells will produce many of them, so they can flood the body with these antibodies and really saturate and let them go out and find the correct antigen. So it's a very effective way for the body to find the antigens that it wants to neutralize by secreting these antibodies, letting them saturate throughout the body, and essentially once they're secreted, they don't need to be part of the B cell anymore. They can go out and bind anywhere in the body. Now, just to contradict that, some of these antibodies, and I'll mention the specific type, actually do stay attached to the B cell. So this is more of the exception to the rule. Now, when the antibodies bind, they're going to make a lock and key fit like this here. I need to actually rearrange this. Let me come back to this one here, because these two aspects here I want to talk about here in a minute. So what are the antibodies actually doing? There's 4 primary things that they do, and these are referred to as opsonization, agglutination, neutralization, and lysis, which lysis is essentially complement. And we talked about that in chapter 14, where the antibodies, or we talked about proteins, but there's a process by which the body punctures holes in the membranes of cells, like bacterial cells. And so antibodies actually play a role in that, and they increase complement. So lysis is in reference to complement. So let's go back to opsonization here. Opsonization is shown here in these first 2 portions of this figure, so here and here. What happens first is the antibodies are going to coat a microbe like a bacteria. This is going to be most commonly directed towards bacteria. So here's our bacterial cell, it's a bacilli. Here's a bunch of antibodies, and they have specificity for the antigens on the surface of this bacteria. So they're binding and they're sticking. Now, opsonization follows where this binding here allows macrophages to grab a hold of these bacteria and phagocytize them more easily. So macrophages can do this anyway, but when they're bound with antibodies like this, it basically signals the macrophage to do it at a faster, more efficient rate. So this, as it says here, they encourage the uptake of the microbe by phagocytosis, like putting handles on a slippery object to provide phagocytes a better grip. So by tagging them, they essentially get phagocytized more effectively and more readily. So it really just increases that process that's already happening anyway. And it's one of the ways, if you remember from chapter 14, it talked about the immune system being redundant, having lots of extra features that maybe aren't totally necessary, but make everything work more effectively. And this is one of those. So it makes that more effective. Another way antibodies have an effect is on the action of viruses. And this is key in the viral infection. What the antibodies are going to do, so going back here, this is neutralization. Apologize, I skipped agglutination. Let me come back to that. Neutralization is shown here. And what's happening is the antibodies are going to bind to the viral spikes. And when they bind to those viral spikes, they're going to block those spikes from being able to attach to the surface receptors of the host cells that they want it to bind to. So essentially, you're blocking the virus from being able to attach to the cell. We talked about viruses having lock and key specificity with the host. Viruses, their spikes being the keys. So if these are the keys, think of the antibodies as putting a piece of gum over the key. So now it can't fit into the lock anymore, and so it's blocked. And that's neutralization. Now, just to connect this back, the way the body fights viruses starts with the T cells, and the T cells are going to clear the infected cells, and that makes it so that the body is not generating more viruses. But the viruses that have already got out of the first cells are now floating around, and so the way to stop them from going on to infect further cells is to neutralize them. So the body fights viruses by eliminating the infected host cells and then blocking the viruses that are floating around the body. Agglutination is most effective against bacteria, and it involves antibodies attaching to 2 bacterial cells with each arm and then locking them together so that they can't move apart. And this happens in a series where lots of bacterial cells are locked together by the cross-linking of the antibody arms against each microbial cell. And this is one of the ways that having 2 arms like this is really, really effective. So this, you can see how that makes this work here. So essentially you get a bunch of antibodies, and they're cross-linking different cells, and they start to link all these together and lock them together. And we call that an agglutination reaction, in which all these cells become clustered, and they can't spread apart, and they can't move anymore. And this is one of the ways the body contains an infection, preventing microbes from becoming septic, for example, traveling throughout the bloodstream. So that's a pretty important reaction that the body has against bacteria. We also talked about lysis, and again, this is in reference to complement. And antibodies essentially assist in the complement process. So by being tagged, complement proteins are more likely to bind, and that increases that process, making that more effective. So that's essentially part of the complement process. Okay, and this last one down here is showing how neutralization can also apply to toxins. So I showed neutralization here where viruses are neutralized. That can also apply to some of the toxic enzymes or toxins that microbes produce. So here it's showing how an antibody can block a toxin and prevent it from having an interaction with the host. So that's another way that neutralization can occur. Okay, so just go back one more time here in case you didn't get it all. These are the 4 primary functions of antibodies. Now, next we're going to get into different types of antibodies. And we have 5 primary types, and they're referred to as Ig within a letter, arbitrary letter, designating the different forms. So Ig stands for immunoglobulin, and then you have immunoglobulin G, immunoglobulin A, immunoglobulin M, D, and E. So you can abbreviate each of those as IgG, IgA, etc. So this is an overview figure, and it's got the shapes of all 5, and it's got a little information about each of these here. I'm not going to really get into too much of this information, mostly down here on the function, but this is a good reference figure to look at them all simultaneously. Now, just real quick, you'll notice most of these, they all actually have a Y shape to them, Y, Y, Y. But some of these here in the middle, IgA and IgM, they also have the Y, but they're arranged in a more complex form. So what ends up happening with these is it conjoins multiple antibodies together and locks them into a more complex shape, but it still retains that basic Y receptor configuration. So the most common antibody immunoglobulin is immunoglobulin G. So what you want to know about this one is it's the main antibody, makes up about 80%, and this is the only antibody that's capable of crossing the placenta. Kind of a random thing. There's lots of information about each of these, but these are kind of the main points of this one. And why this is key is because placental immunity is somewhat limited during the development process. So when the fetus is developing inside the mother's womb, inside the placenta, they're getting protection from the mother, but it's somewhat limited. And part of that limitation involves really only one antibody being able to cross. And while it's an important one, that does leave the developing child just a little bit more susceptible than it would be otherwise. So recognize that IgG is the only one capable of crossing the placenta. IgA is the one that we showed back here with what's called a dimer form. And a dimer is essentially 2 of the same molecule joined together in some kind of mirror image. So imagine 2 molecules staring at each other in a mirror, and they're linked together at the same time. And that's really what a dimer is. And a dimer is a term that's used in many different contexts, not just antibodies. So IgA is the only one to form a dimer. It can also stay as a monomer, meaning just the one, not bound together. But commonly it's in a dimer form. And so you want to recognize that. And then the other thing is that this one is really important in mucosal defenses. And really that's more important. The dimer form is somewhat of a physical thing. This is more of a physiological thing. IgA is going to get saturated in your mucosal layers. So remember we talked about mucus back in chapter 14, even chapter 13, and we described it as a defense mechanism from the first line. It's a barrier. So it traps microbes. It's a way to flush them out. But one of the things that happens over time, as you're exposed to these microbes more and more, and you produce these antibodies against them, within the mucus you start to saturate IgA antibodies. And they're going to have specificity to what you've been exposed to previously. So as these microbes come back and visit our bodies over and over, that mucus is going to get more and more potent against them because it's going to have IgA saturated within it. And if we've seen those microbes before, we're going to have IgA antibodies against them. So it gets harder and harder for them to bypass that mucus, and that makes our mucus more and more effective over time. So IgA plays a big role there. Now, just to make a quick note about this dimer form, this is significant because it allows more effective agglutination and cross-linking. So now we have a molecule with 4 arms instead of just 2, and that cross-linking and agglutination is even more effective. And guess what? This one here, which I'm going to talk about next, is the king of agglutination. It's got 10 arms instead of 4, so it's even more effective. Okay, so that one, by the way, is IgM. So this is the pentamer. It's got 5 of those Y-shaped antibodies attached together, and this is, again, the king of agglutination right here. So IgA and IgM are both really good at it, but this one is superior. And so you want to recognize this is the one with the pentamer. That pentamer shape makes it good for agglutinating. And the other thing is that this is the first antibody made during infection. And let me just go back and connect that to one of the figures here. This one here. And this was showing the activation of the B cells. Here's the plasma cell making the antibodies. And if you notice, this is the first one coming out. Or it's the only one coming out, but it's showing this one because it's indicating that this would be the first one produced. Okay, that's why that figure shows that particular version. So first one produced. It doesn't go on to be the most numerous. That's IgG, but it is actually the first one made, and that's really effective at agglutinating and trapping bacterial cells together. IgD and IgE are they're much less common. They're produced in much smaller quantities. But IgD is the one that acts as the receptor. So I mentioned earlier that in some cases, go back here just to specify, it says some are expressed on the surface of B cells. They serve as B cell receptors, talking about different types of antibodies and immunoglobulins. So to clarify, that is specifically IgD. So IgD is the one that acts as a receptor. So it's actually not secreted, and it's the exception to the rule here. But otherwise it does the same thing, it just acts as a receptor. So IgE then, the least common of all of them, this one is an interesting one. It has a role in relation to allergies, parasitic worm defenses, and it interacts with some of the cells from our second line of the immune system, which are basophils and mast cells. Now, this is discussed in much greater detail of chapter 16, which is disorders of the immune system, which I'm not going to get into. I would encourage you to read it. It's very interesting, and IgE goes back to the allergy worm connection and the whole way that the body deals with those in a similar way. But all you need to know for now, and for this class really, is that IgE is the antibody that is involved in allergic reactions and parasitic worm defenses. So it's unique in that sense. Okay? So those are the 5 antibody types there. Now the next thing is in reference to the production of antibodies. Antibody production is going to go through 2 general distinct phases. And this goes back to what I put on the board the other day about the delayed response between the first time you're exposed and the time your body actually produces any effector cells. You know what, let me just pause this and put this up here on my paint document so you can make sure we're all on the same page here. Just a second. Okay, so this is what it looked like. I was talking about a naive cell, in this case a naive B cell, so they've not yet been activated. They're experiencing the antigen for the first time. And what's going to happen is that they're going to go through the expansion, and they're going to produce memory cells and effector cells. But the first time this happens, there's a delay, and it doesn't take place very quickly. And again, I said that this leaves the host susceptible. So we're susceptible during this time period of delay. Now, as I said, after we're exposed, and once we get exposed in the second time, what's going to happen is we activate memory cells, which transition directly back into effector cells, and we get no delay time in that reaction. So this is what I getting into now in a little bit more detail, and that's what this whole topic here is about. Okay, so the first time we're exposed, we call this the primary response, and it involves a latent period, and that's the delay. So the technical term there is called a latent period. That's that delay. Marks a lack of antibody production because of the delay time. So during a secondary response, we've seen the pathogen already, and now we're exposed to it again. This is what's referred to as an anamnestic response. And what it does is it triggers the memory cells directly, and they respond without the delay. And this involves a much faster type of reaction. So you can, oh, excuse me, I got one more slide, and then I've got the figure here. The idea is that after the latent period, when we get to the secondary or anamnestic response, we have less of a delay, virtually no delay. We actually end up producing more antibody, and the body antibodies persist longer because they are synthesized longer. So the antibodies themselves don't necessarily last any longer, but they are produced over a longer period of time. So they last longer because they're being produced more. And just to clarify, one thing I've never mentioned, I should point out, antibodies have a shelf life of about 4 to 6 weeks, something like that. You're talking about a matter of weeks, maybe a handful of weeks, and then they tend to wear out and have to be replaced. So if you don't continue to produce those, then they will wear out relatively quickly. What makes those last longer is memory cells that are going to produce more and more of them over longer periods of time. So every time we're exposed further, then we produce more antibody. Now, this is showing the difference in antibody production. Titer, by the way, is a term that describes the quantity of antibody in the blood. So a titer is a measure of antibody in the blood. That's what that is. So antibody production goes up here. So, as the line goes up, that's showing an increase in antibody production. And up here, note that we're looking at total antibody production. So all 5, represented by the blue line here, the dark blue. We're looking at IgM only with the red line, so it's showing IgM production, and then IgG is shown with the light blue line here. So you see not only the quantity, but then you're also looking at the time period which is being produced, how quickly and over what type of longevity. So looking back then here at the latent period, we see that upon first exposure, there's really nothing happening during this latent period, which can last for several days in some cases. So there's a relatively long period here where the body really is not doing anything that is effective against the pathogen. It's doing things, but nothing really has amounted to a response yet. So that is the window of opportunity for the pathogens, and typically when you and I start to get sick is during this vulnerable time period. Now, as we reach the end of this, our body starts to produce these antibodies for the first time. It's going to start with IgM, and it's going to get produced in a relatively large quantity, and other antibodies then are going to get produced over time, primarily antibody IgG. So these are going to be the 2 primary ones produced, and we're going to max out within a relatively long period of time between first exposure and maximum production. Okay, so even early on, we're still somewhat susceptible because we haven't maxed out yet. Okay, so that's the first exposure. Now, the other thing about this is that antibody production tends to taper off pretty quickly because the memory cells that produce them have only been somewhat minorly stimulated, so to speak. So they've been stimulated, and the memory cells are going to persist for a long time. But the antibodies they produce are essentially, the antibodies produced by the plasma cells, rather, are going to be relatively short-lived. So antibody production takes a while to get started, and it doesn't last very long. Now, secondary exposure to antigen, the anamnestic response, if you note, what's going to happen is as soon as we're exposed, and this by the way involves direct binding to the memory cells, the memory cells then are going to immediately spring into action, and they're going to differentiate into plasma cells, and they're going to immediately start producing large amounts of antibody. So note that from the second exposure, you have an almost immediate max production of antibodies, and there's virtually no delay in that. It just immediately kicks right into antibody production. So we have virtually no delay period. And this is why we don't see infection happening over and over and over from the same types of microbes, because our body just kicks right into a response without any delay. Now, the other thing is that the memory cells are going to produce antibody longer, or the memory cells are going to trigger effector cells. Excuse me, I keep saying that wrong. Essentially, they're going to generate antibodies over a longer period of time. And this really what this has to do with is the memory cells differentiating into plasma cells, which are going to increase the antibody production. So, essentially, faster response and longer duration of the antibody in the blood. Okay, now every time you're exposed thereafter, this theoretically keeps going up and up and up. So if we had another additional room over here, we would see that a third exposure, you'd have even more antibody production, and it would last even longer. And this is kind of the idea behind booster shots. As we'll look here at the very end, a booster shot, or a vaccine, is an artificial way of triggering the body into a response. And a booster shot is essentially the equivalent of a secondary exposure. So your first vaccine that you get is your primary response. And sometimes that's all you need. Excuse me, I keep hitting the mic. Sometimes that's all you need. You just need the body to see it once, and then if you're exposed to it again, you immediately kick into the secondary anamnestic response. So that's the idea of a vaccine. Trick the body into having a primary response without any real threat. And then you go through all of this without any real harm, and you're ready for it upon a secondary exposure. Now, if you're exposed to the real virus or whatever it is you're vaccinated for, your body has an immediate response, and there's no chance for the actual pathogen to do any harm. But a booster shot is going to take that even further, and that is going to increase antibody production even higher. So if you get a second shot, for example, a second vaccine, then that artificially elevates the antibodies even more. So now the third exposure, if that's the real one, you'd have an even higher concentration of antibodies than you would after the first. And in some booster shots, you keep going and you do a third booster shot, and each time you're just boosting more memory cells, more antibody, and more of a defense against these pathogens. And for little children who don't have really any immunity yet, that can be important to give them as many antibodies as research has indicated that they could possibly have to fight off these infections. And that's the idea behind vaccinating and booster shots. Trick the body out of the primary response, get it ready for a secondary response, and then sometimes get it ready for an even more exaggerated response by having 2 or 3 booster shots. So this is where we get into different types of immune responses. There are two general types called active and passive. Active is everything that we've just talked about in terms of stimulating things the natural way. So in this case, an active immunity leads to memory cells, and we have the delayed response in the first line. So essentially, active is the natural way that it happens. Let me contrast that with the other type that we haven't talked about yet, and that is called passive immunity. So passive immunity is the exception to the norm. And in a passive immunizing event, what happens is that an individual can gain the protection of antibodies for a short period of time. What happens is you can donate someone's antibodies to another individual. And interestingly, the antibodies themselves, and this is key, the antibodies themselves actually don't have self versus non-self specificity. In fact, let me just write that here. Antibodies don't have self versus non-self specificity. Now that's not on the study guide, but why that's key is because this allows people to donate antibodies back and forth. And the antibodies will work the same outside of your body as they would inside of your body. And that's the idea behind passive immunity, is that you can essentially donate them and let someone else use them. The difference between passive and active is that when you donate antibodies, you don't generate memory cells which actually produce them. So the memory cells are what create the plasma cells which go on to make the antibodies. So essentially, when you do a passive immunizing event, you're just letting someone borrow the antibodies, and they can't actually make them themselves. So the trade-off is that they only last as long as the antibodies last, which, as I mentioned, is a handful of weeks. So you only get antibody persistence over a short-term duration of time, and you don't make any antibodies. So once they go away, and you have to have another donation for them to persist. Okay, so active is essentially the ability of the body to generate its own memory cells and its own antibodies, and then you get long-term protection because of the memory cells. Okay, so one way to think about this is active equals memory cells. That's the key thing. And then that gives you long-term protection. The passive, you have no memory cells, and as a result, short-term protection. And just to add to that, passive almost always involves antibody donation. And we'll talk about the different ways that that can happen here in a second. So active versus passive. Now, another way, and this is a typo by the way, I'm sorry, this should say natural versus artificial. Take that word out and replace it with artificial. So in addition to being active and passive, memory versus no memory, immunizing events can also be natural and artificial. Natural is, once again, everything that we've just talked about, in that it happens through normal exposure, and the body has to respond given a natural ability to recognize the antigen and trigger the third line of the immune system. So natural means natural. Artificial is basically any sort of clinical or medical context in which you're tricking the body through, I'm trying not to use the word artificial, you're tricking the body through a fake mechanism of thinking it's been exposed to the antigen. I'm not doing a very good job of articulating that. But let me try a little bit more here. We can combine some of these together, and we can have, for example, natural active. So natural active is, once again, the kind of most common general way that the body is going to activate an immune response, and this is everything that we've talked about. So it's natural in that you're exposed to pathogen through everyday experiences, and then it's active in that it triggers long-term immunity. So when you're exposed to a virus by the person sitting next to you and you get sick from that virus, you have natural exposure through everyday experience, and that is going to activate long-term immunity, and that's natural active. So by far the most common. Natural passive is very specific. So it's natural in that it occurs through natural exposure. It's passive in that it's short-term. And really the only way this actually happens is during pregnancy between a mother and a developing child. That is the example. Now there's two different examples within, and it can happen both prenatally in the womb and postnatally outside the womb. Prenatally involves the transmission of IgG across the placenta. So if you go back and look at IgG, I said it's the only one that crosses the placenta. This particular antibody IgG then is responsible for prenatal natural passive immunity. And what that means is that the mother is going to donate antibodies into the developing fetus, into the placenta, and give that developing child a prenatal natural passive immunity. Now, when the child is born, postnatally, this involves breast milk. And breastfeeding is going to have antibodies, or excuse me, breast milk is going to have antibodies in it, and breastfeeding then is going to naturally, and from a natural passive standpoint, pass those antibodies into the child and allow the body to use those for short term. So breast milk and breastfeeding is really key. Everybody kind of knows that, I think. But one of the key things that breastfeeding does is it gives the child a natural passive immunity. And when children are breastfed for the first so many months or years of their life, they're getting antibodies throughout that entire time period. Now, antibody production is actually higher early on, and a milk called the colostrum actually has more antibodies in it early on than it does later on. Okay, so that's the idea of natural active and natural passive. Now, artificial active is a way in which you can trick the body into developing memory cells. And the example here is a vaccination. Really, a vaccination is artificial active immunization. So what you do with the vaccine is you take a little piece of the pathogen and you separate that from the entire microbe. So you're basically going to kill a microbe, chop it into little tiny pieces, and then inject that into the body so that you only have the antigens floating around, not in the intact form. So recall that our body recognizes the antigens of a microbe, not the microbe as a whole. So when you chop the microbe up and you inject it, you get the antigens from the microbe without the microbe being active and alive and harmful. And all your body has to do is see little pieces of the microbe and it will generate antibodies and memory cells against it without actually any real threat from the microbe being real. So that's the idea of a vaccine, and that's considered artificial active. It's artificial because we're simulating a real event. It's not an actual exposure, but it's considered active because we do get long-term memory, and we create memory cells in that process. The body doesn't know that it's artificial. It responds just as if it was real. So we get the same long-term protection as if we were actually exposed. And one of the reasons why you actually get sick sometimes, so, quote unquote, when you get a vaccine, is because of the way your body responds to infection. The body is going to respond to infection with inflammation, fever, headache, etc. Those are all part of the natural response. And when it doesn't know the difference between the real thing and the fake, it responds the exact same way. So it may not be the same intensity of infection, and it shouldn't. Most time you won't get sick at all, but sometimes you get kind of sick and you can definitely get run down. That's the natural way your body responds. So whether it's real or not real, you tend to respond the same kind of way for the most part. Now, there is a difference, and everybody responds a little differently to a vaccination. But that's one reason why people get sick, is it's really just the body's response that actually makes you feel that way, not so much the pathogen. The difference is that the pathogen is not going to do any damage with the vaccine. So you're only dealing with the body's response, which, if you recall from Chapter 13, is the indirect damage that pathogens induce. So there's no direct damage, but the indirect damage can be just as in some cases just as bad as what it would be otherwise, and it certainly can be noticeable. And that's why people sometimes will claim to get sick after a vaccine. Not really getting sick; they're experiencing the symptoms that your body goes through during an infection, and many of those are self-induced and have nothing to do with the direct activity of the pathogen. Okay, so that's an artificial active. Artificial passive goes back to the idea of donating antibodies. And in this situation, what's happening is the antibodies are going to be artificially transfused from donor patients. And the idea is that in a medical setting, researchers and doctors and physicians and everyone involved can actually extract antibodies from donor patients. Those antibodies can then be concentrated and then injected via transfusion into a patient who needs them. And that's the idea of artificial passive. And in today's world, researchers can stockpile antibodies against a variety of common illnesses. And then a patient can essentially purchase those antibodies and have them transfused into their body. And this would be done for people who are immune compromised. Cancer patients, organ transplant victims, HIV, people suffering from HIV, and there's a whole host of different types. But if you're immune compromised, that means that you can't produce the antibodies on your own, or at least not to a sufficient level. So because you can't do that on your own, then in a medical context, you can have antibodies given to you, and they will give you short-term protection as long as they last. Again, the trade-off is they don't last long-term. So if you're getting transfusions of antibodies, you're going to have to get that every so many weeks, and you're going to have to go back and continually receive that transfusion. So it's a way to kind of artificially give someone antibody protection without, when they can't do it themselves. Okay, so here's a figure showing these 4 different examples. So active, you get sick and you're just dealing with the infection. Natural, active, natural, passive, you're looking at, the mother transfusing antibodies to the child. This would be a postnatal. It can also happen prenatally. Active involves vaccination. This would be artificial active. Really is a vaccination. And then artificial passive is the transfusion of antibodies into a donor. So you can see that here. They've got that in the IV, and they're transfusing that into her arm here. So she looks like a cancer patient, someone who would have a low white blood cell count and would need that sort of transfusion. So that's what's happening there. Okay? I'm going through this kind of fast. I apologize if I went too fast. Please email me if you have any questions, and I'd be happy to answer those. I'll try and get back to you as soon as I can. Okay? So have a great week, and I will see you all next Wednesday after the election. Goodbye.